1 in every 100 people has a condition where their own immune system attacks their intestines every time they eat bread. 83% of them don't know it. On average, it takes 6 to 10 years from the first symptoms to get a diagnosis. And during that time, while doctors run tests and rule out other conditions and suggest stress management, something else is happening inside the body. The immune system is learning, adapting, and in some cases, it starts attacking more than just the gut. Celiac disease has long been understood as a digestive disorder. Eat gluten, damage your intestines, feel terrible. Remove gluten, heal, feel better. Simple. But over the past 2 decades, researchers have uncovered something far more complicated and far more consequential. Celiac disease isn't just about digestion. It's an autoimmune condition. And it clusters with other autoimmune conditions in ways that are forcing scientists to rethink how these diseases develop in the first place. This is the story of a protein, a lock, and a case of mistaken identity that may explain why autoimmune diseases are rising around the world. Gluten is a family of proteins found in wheat, barley, and rye. It's what gives bread its elasticity, pasta its chew, and baked goods their structure. Humans have been eating gluten for at least 10,000 years, since the agricultural revolution first domesticated these grains. For most people, gluten is unremarkable. It's digested like any other protein, broken down into amino acids, absorbed, used, forgotten. But for a significant subset of the population, gluten is a trigger, the starting point of an immune response that can range from mild discomfort to severe, systemic inflammation. There are 3 main conditions associated with gluten, and they're often confused with each other. Celiac disease is an autoimmune disorder. When someone with celiac eats gluten, their immune system attacks the lining of the small intestine, specifically the villi, tiny finger-like projections that absorb nutrients. Over time, these villi flatten and degrade, leading to malabsorption, nutritional deficiencies, and a cascade of downstream effects. Celiac is genetic. You need specific variants of genes called HLA-DQ2 or HLA-DQ8 to develop it. About 30 to 40% of the population carries these genes, but only about 1% actually develops celiac disease. Something else has to happen. An environmental trigger. A moment when the immune system tips from tolerance into attack. NCGS is more mysterious. People with NCGS experience many of the same symptoms as celiac patients: bloating, abdominal pain, fatigue, brain fog. But when doctors perform a biopsy, there's no villus atrophy, no autoimmune destruction. For years, many physicians dismissed NCGS as psychosomatic, a nocebo effect driven by diet culture and food anxiety. That's changing. Recent research has shown that NCGS involves a distinct immune response, different antibodies, different inflammatory markers. It's real. It's measurable. And it may affect 1 to 2% of the population, roughly the same prevalence as celiac disease itself. Wheat allergy is different from both. It's a classic allergic reaction, mediated by IgA antibodies, the same type involved in peanut allergies or hay fever. Symptoms can include hives, difficulty breathing, and in severe cases, anaphylaxis. Wheat allergy is most common in children and often resolves with age. Celiac disease affects an estimated 1 in 100 people worldwide. In the United States alone, that's over 3 million people. But here's the problem: most of them don't know it. Studies suggest that up to 83% of Americans with celiac disease are either undiagnosed or misdiagnosed with other conditions: irritable bowel syndrome, chronic fatigue, anxiety, anemia of unknown origin. The average time from first symptoms to diagnosis is 4 to 6 years. Some studies put it even higher: 6 to 10 years of uncertainty, of being told nothing is wrong, of symptoms that wax and wane without explanation. This delay isn't just frustrating. It's dangerous. Research has shown that the longer celiac disease goes undiagnosed, the greater the risk of developing additional autoimmune conditions. One landmark study found that children diagnosed before age 2 rarely developed other autoimmune diseases. But those diagnosed after age 20 had a significantly elevated risk for thyroid disease, for type 1 diabetes, for conditions that would follow them for the rest of their lives. The immune system, it seems, has a memory. And the longer it practices attacking self, the better it gets. The gut barrier. Your intestines are not just a passive tube. They're a highly selective barrier, a border crossing that decides what gets into your bloodstream and what stays out. The cells lining your small intestine are bound together by structures called tight junctions. Think of them like the seals between tiles in a bathroom. They keep water from leaking through to the subfloor. In your gut, tight junctions keep bacteria, toxins, and undigested food particles from slipping between cells and entering circulation. When tight junctions are working properly, only fully digested nutrients pass through, absorbed directly into the cells, processed, and released into the bloodstream in forms the body recognizes and can use. When tight junctions fail, larger molecules slip through the gaps. Partially digested proteins, bacterial fragments, things that don't belong in the blood. This is what researchers call increased intestinal permeability. You might know it by its more colloquial name, leaky gut. For a long time, leaky gut was considered fringe science, the domain of alternative medicine practitioners and wellness influencers. Mainstream gastroenterologists were skeptical. The gut barrier was supposed to be stable, its permeability fixed. Then, in 2000, a researcher named Alessio Fasano discovered zonulin. Zonulin is a protein produced by the cells of the intestinal lining. Its job is to regulate tight junctions, to open them when necessary and close them when not. It's the key that unlocks the door between the gut and the bloodstream. In healthy people, zonulin is released in small amounts, in controlled circumstances. The door opens briefly, then closes. But Fasano and his team discovered something crucial: certain triggers cause zonulin to spike. And when zonulin levels stay elevated, tight junctions remain open. The door stays unlocked. Two triggers stood out as the most powerful stimulators of zonulin release. The first was bacteria, specifically, an overgrowth of bacteria in the small intestine, where they don't normally belong in large numbers. The second was gluten. The gluten-zonulin connection. When gliadin, a component of gluten, reaches the small intestine, it binds to a receptor called CXCR3 on the surface of intestinal cells. This binding triggers a signaling cascade that releases zonulin. Here's the remarkable part: this happens in everyone. Not just people with celiac disease. Not just people with gluten sensitivity. Everyone. Gluten triggers zonulin release in all humans. The difference is magnitude and duration. In healthy individuals, the zonulin spike is brief and the tight junctions reseal quickly. In people with celiac disease, zonulin levels rise much higher and stay elevated much longer. The door doesn't just open, it stays open. Fasano's research suggested something provocative: the body treats gluten like a microbial threat. The zonulin pathway evolved as a defense mechanism, a way to flush out harmful bacteria by temporarily increasing intestinal permeability. Gluten, for reasons not entirely understood, activates this same pathway. The body mistakes it for something dangerous. When tight junctions are compromised, the consequences extend far beyond the gut. Bacterial fragments, particularly lipopolysaccharides from gram-negative bacteria, enter the bloodstream, triggering systemic inflammation. Partially digested food proteins slip through, provoking immune responses. The body begins producing antibodies against substances it was never supposed to encounter directly. This creates a feedback loop. Inflammation damages the intestinal lining further. More zonulin is released. More material leaks through. The immune system becomes increasingly activated, increasingly vigilant, increasingly prone to error. And errors, in the immune system, can become permanent. The immune system operates on pattern recognition. It learns to identify threats by their molecular signatures, specific shapes, specific sequences. When it encounters something new, it catalogs it, files it away, prepares a response for next time. When undigested proteins and bacterial fragments leak into the bloodstream, the immune system doesn't know they came from food. It doesn't know they're harmless. All it sees are foreign molecules where no foreign molecules should be. So it attacks. It produces antibodies. It activates inflammatory pathways. It does exactly what it's supposed to do when facing an invader. The problem is that some of these leaked molecules look, to the immune system, remarkably similar to the body's own tissues. This is where the story takes its darkest turn. The immune system's memory. Your immune system is a learning machine. Every pathogen you've ever encountered, every virus, every bacteria, every foreign protein, has left a trace. Your body remembers them. It is cell-specifically trained to recognize them, antibodies pre-formed and waiting. This is why vaccines work. Expose the immune system to a harmless version of a threat, and it builds a defense that lasts years, sometimes decades, sometimes a lifetime. But this memory system has a flaw. It relies on pattern matching. And pattern matching can produce false positives. Molecular mimicry is the term for what happens when two different molecules share structural similarities, similar enough that the immune system can't tell them apart. Here's how it works. Your immune system encounters gluten, specifically, gliadin peptides that have leaked through a compromised gut barrier. It recognizes these peptides as foreign. It mounts an attack. It produces antibodies designed to target and destroy anything with that molecular signature. So far, so good. That's exactly what the immune system is supposed to do. But gliadin, it turns out, shares structural features with several human tissues. The protein sequences aren't identical, but they're close enough. Similar shapes. Similar binding sites. Similar enough to confuse an immune system already primed for attack. The antibodies meant for gluten start cross-reacting with the body's own cells. This is autoimmunity. Not a malfunction, exactly, more like friendly fire. The immune system is doing its job. It's just been given the wrong target. The most well-documented example of gluten-related molecular mimicry involves the thyroid gland. Gluten proteins share structural similarities with thyroid tissue, specifically, with the enzymes and receptors that regulate thyroid function. When the immune system produces antibodies against gluten, some of those antibodies also attack the thyroid. This connection helps explain why autoimmune thyroid diseases, Hashimoto's thyroiditis and Graves' disease, are so common in people with celiac disease. Studies have found that people with celiac have significantly elevated rates of thyroid autoimmunity. In some populations, the overlap is striking. Up to 30% of celiac patients have thyroid antibodies. And here's what's particularly troubling: the connection works in both directions. People with autoimmune thyroid disease have higher rates of celiac disease than the general population. The conditions cluster. They feed each other. Some researchers have proposed that for genetically susceptible individuals, gluten may be the initial trigger, the spark that ignites a broader autoimmune cascade. Remove the gluten, remove the trigger, and the fire may eventually burn itself out. This isn't proven. The research is still evolving. But there's enough evidence that some clinicians now recommend gluten-free diets for patients with Hashimoto's, even in the absence of diagnosed celiac disease. The autoimmune cluster. Thyroid disease isn't the only condition that clusters with celiac. The list of associated autoimmune disorders is long and growing. Type 1 diabetes, multiple sclerosis, rheumatoid arthritis, Sjogren's syndrome, Addison's disease, autoimmune hepatitis, dermatitis herpetiformis, pernicious anemia. The connection to pernicious anemia is particularly instructive. Pernicious anemia is an autoimmune condition where the body attacks the cells in the stomach that produce intrinsic factor, a protein necessary for absorbing vitamin B12. Without intrinsic factor, B12 deficiency develops, leading to anemia, neurological problems, and fatigue. Pernicious anemia shares genetic risk factors with celiac disease. Both involve the HLA genes that shape immune function. Both involve autoantibodies attacking specific tissues. And both are more common in people who have the other. Research has shown that patients with pernicious anemia have elevated rates of celiac antibodies. Screening for celiac disease is now recommended for anyone diagnosed with pernicious anemia, and vice versa. The pattern is consistent: autoimmune diseases don't occur in isolation. They cluster. They share genetic architecture. They share environmental triggers. And increasingly, researchers believe they share a common pathway, one that begins with a compromised gut barrier and ends with the immune system at war with itself. The time factor. Remember that study about diagnosis timing? The one that found children diagnosed early rarely developed additional autoimmune conditions, while those diagnosed late had elevated risk? The implication is sobering. Every year that celiac disease goes undiagnosed is a year of continued intestinal damage. A year of leaky gut. A year of molecular mimicry. A year for the immune system to make new mistakes, to find new targets, to build new autoantibodies. For decades, non-celiac gluten sensitivity occupied an uncomfortable position in medicine. Millions of people reported symptoms after eating gluten, but their tests came back negative. No villus atrophy. No celiac antibodies. No measurable pathology. Many doctors concluded that NCGS wasn't a real condition, that patients were either misattributing symptoms to gluten or experiencing a nocebo effect, where belief in harm creates the perception of harm. This dismissal was understandable, in a way. Medicine relies on measurable markers. If you can't biopsy it, image it, or detect it in blood work, it's hard to prove it exists. But the patients kept coming. And they kept getting better when they stopped eating gluten. The Columbia Study. In 2016, a research team at Columbia University published a study that changed the conversation. Led by immunologist Armin Aladini, the team examined 80 individuals with NCGS, comparing them to 40 people with celiac disease and 40 healthy controls. All participants were eating unrestricted diets containing gluten when they joined the study. The researchers measured several markers, including subclasses of immunoglobulin G, the antibodies produced by B cells in response to dietary proteins. What they found was striking. Patients with celiac disease showed elevated levels of IgG1 and IgG3, the earlier, more inflammatory forms of immunoglobulin. These antibodies appeared alongside the extensive gut damage characteristic of celiac. But patients with NCGS showed a different pattern. They had elevated IgG2 and IgG4, later stage, less inflammatory antibodies that typically appear as the immune system winds down a response. In other words, NCGS and celiac disease produce distinct immune signatures. They're not the same condition. They're not even the same type of inflammation. What this means? The Columbia findings suggest that NCGS is real, measurable, and biologically distinct from celiac disease. People with NCGS aren't imagining their symptoms. They're experiencing a genuine immune response to gluten, just a different kind than celiac patients experience. The elevated IgG4 in NCGS suggests that the immune system is attempting to regulate itself, to cool down an inflammatory response. But the elevated intestinal permeability marker FABP2, which was also found in NCGS patients, indicates that gut damage is still occurring. The damage is subtler than in celiac disease. It may occur in a different part of the intestine. It may not be visible on a standard biopsy. But it's there. As Aladini put it, sensitivity to wheat in the absence of celiac disease is a condition distinct from celiac disease, with a biological basis. The problem is that we still don't have a validated blood test for NCGS. The diagnosis remains one of exclusion: rule out celiac, rule out wheat allergy, try a gluten-free diet, see if symptoms improve. This means many people with NCGS are never formally diagnosed. They self-identify as gluten sensitive, avoid bread and pasta, and feel better, but without medical confirmation, they're often dismissed or doubted. The Columbia research points toward a possible future, a test that measures both IgG4 antibodies to gluten and FABP2, the marker of intestinal damage. Together, these could help confirm NCGS in patients who've already tested negative for celiac. But for now, the gray zone remains. Millions of people in it, navigating a medical system that's only beginning to acknowledge their condition is real. How many people actually have NCGS? Estimates vary widely. Some researchers put it at 1% of the population, similar to celiac disease. Others suggest it could be as high as 6%. The uncertainty reflects the diagnostic challenge. Without a definitive test, we can't conduct proper prevalence studies. We're left with self-reported data, elimination diet trials, and educated guesses. What we do know is that gluten-free diets have exploded in popularity over the past 2 decades, far outpacing the diagnosed celiac population. Some of this is driven by fad dieting and health trends. But some of it, almost certainly, reflects real but undiagnosed gluten sensitivity. The science is catching up. Slowly. The only treatment. As of today, there is exactly one proven treatment for celiac disease: a strict, lifelong gluten-free diet. No medication. No procedure. No shortcut. The diet must be absolute. Even small amounts of gluten, as little as 50 milligrams, roughly 1/64th of a slice of bread, can trigger intestinal damage in people with celiac disease. Cross-contamination from shared cooking surfaces, fryers, or utensils is enough to cause problems. This is harder than it sounds. Gluten is everywhere. It's in obvious places like bread, pasta, and beer. But it's also hidden in soy sauce, salad dressings, medications, supplements, and processed foods of all kinds. It's used as a binding agent, a stabilizer, a filler. Reading labels becomes a daily practice. Eating out becomes a negotiation. For people with celiac disease, this isn't a lifestyle choice. It's a medical necessity. The alternative is continued intestinal damage, malabsorption, and escalating autoimmune risk. The good news is that the gut can heal. Once gluten is removed, the intestinal lining begins to repair itself. Villi regenerate. Nutrient absorption improves. Symptoms resolve. For most people, significant healing occurs within the first year of a gluten-free diet. Complete mucosal recovery may take 2 to 5 years, depending on the severity of damage at diagnosis. Nutritional deficiencies, iron, B12, folate, vitamin D, calcium, typically normalize as absorption improves. Many people report dramatic improvements in energy, cognition, and overall well-being. But healing isn't guaranteed. A subset of celiac patients, perhaps 5 to 20%, continue to experience symptoms and intestinal damage despite strict gluten elimination. This is called non-responsive or refractory celiac disease. Some cases result from hidden gluten exposure. Others reflect permanent damage or additional conditions that have developed alongside celiac. Researchers are working on alternatives to the gluten-free diet, medications that could protect the gut from gluten's effects or even induce tolerance. The most advanced candidate is liraglutide acetate, a drug designed to inhibit zonulin. By keeping tight junctions sealed, liraglutide could theoretically prevent the increased intestinal permeability that initiates the autoimmune cascade. Clinical trials have shown promise. Liraglutide reduced symptoms and intestinal inflammation in celiac patients exposed to gluten. It's currently in phase 3 trials, the final stage before potential FDA approval. Other approaches are being explored: enzymes that break down gluten before it reaches the intestine, vaccines that induce tolerance, biologics that modulate the immune response. None of these are ready for clinical use. The gluten-free diet remains the standard of care. But for the first time, there's a real research pipeline, a possibility that future celiac patients might have more options than diet alone. The treatment for non-celiac gluten sensitivity is also gluten avoidance, though the threshold may be different. Some people with NCGS can tolerate small amounts of gluten without symptoms. Others are as sensitive as celiac patients. The variation reflects the fact that NCGS is likely not a single condition but a spectrum of related sensitivities. Unlike celiac disease, NCGS may not be permanent. Some research suggests that after a period of strict avoidance, during which intestinal permeability normalizes and inflammation resolves, some people with NCGS can reintroduce small amounts of gluten without problems. This isn't guaranteed. And without reliable biomarkers, there's no way to know in advance who might recover tolerance and who won't. For now, the practical advice is similar to celiac: avoid gluten, see how you feel, adjust based on your body's response. The research on gluten and autoimmunity points toward a broader truth about health and disease. The gut is not just a digestive organ. It's an immune organ. 70% of your immune cells reside in and around the intestines. The barrier between gut contents and bloodstream is the front line of immune defense. When that barrier fails, the consequences extend far beyond digestion. Systemic inflammation, autoimmune activation, diseases that seem to have nothing to do with the gut. This is the new frontier of medicine, understanding how intestinal permeability, the microbiome, and diet interact to shape immune function throughout the body. Gluten sensitivity, whether celiac or NCGS, is one window into this larger system. The questions being asked now go beyond any single protein or any single disease. Can autoimmune diseases be prevented by protecting intestinal integrity early in life? We don't have all the answers. But we're asking better questions than ever before. One protein. One unlocked door. One case of mistaken identity. Celiac disease was first described over 2,000 years ago by the Greek physician Aretius of Cappadocia. He called it koiliakos, suffering in the bowels. For most of history, that's how it was understood: a digestive disorder, an affliction of the gut. We now know it's much more than that. Celiac disease is a window into how the immune system can turn against itself, how a single dietary trigger, in genetically susceptible people, can initiate a cascade that spreads far beyond the intestines. And celiac may be just the beginning. The mechanisms being uncovered, zonulin, tight junction dysfunction, molecular mimicry, appear to play roles in conditions we never associated with diet or gut health. Type 1 diabetes, multiple sclerosis, thyroid disease. The autoimmune epidemic of the 21st century may have roots in the gut. None of this means everyone should avoid gluten. For most people, gluten is harmless, a routine protein, unremarkable and unproblematic. But for those who react to it, the 1% with celiac disease, the 1 to 2% with NCGS, the undiagnosed millions still searching for answers, understanding what gluten actually does to the body is the first step toward healing. If you've experienced persistent digestive symptoms, unexplained fatigue, anemia that doesn't respond to iron, thyroid problems, or any of the other conditions that cluster with celiac disease, consider asking your doctor about testing. Celiac screening is a simple blood test. It could change your life. And if you've already been diagnosed with celiac, with NCGS, with any autoimmune condition, know that you're not alone. The science is advancing. The treatments are improving. And the medical community is finally recognizing what patients have known all along: this is real. It matters. And you deserve answers.